any heritable change in a gene is known as mutation. Now the question arises, what is the nature of mutations at the molecular level, how it occurs, and how they affect gene and the function of the gene. Although most mutations take place spontaneously, they can also be induced by radiation and a variety of chemical agents that damage nucleotides or that produces breaks in the polynucleotide chain. The agents that are capable of inducing mutations are known as mutagens. Most types of DNA damage are generally corrected by special repair enzymes that are present in the body, and they repair it almost immediately after the mutations occur. The new mutations that are transmitted in any generation therefore represents the minority of changes in DNA that have escaped repair. When can a mutation happen, or what is the frequency of mutation? It is known about mutations that it can happen anytime and in any cell, which means that there is no time specification or no specification for the cells that are vulnerable to mutation. The phenotypic effects of a mutation can range from minor alterations that are detectable only by molecular methods, for example DNA polymorphism, to some drastic changes in essential processes that cause death of the cell or an organism. Mutations can be classified in a variety of ways. Most mutations are spontaneous, which means that they occur in the absence of any cause. Mutation can also be induced through damage to DNA caused by chemical agents or radiations. In multicellular organisms, one important distinction is based on the type of cells in which the mutation first occurs. Those that arise in the cell that ultimately form gametes are termed germline mutations. All other mutations are known as somatic mutations. A somatic mutation yields an organism that is genotypically, and for many dominant mutations, phenotypically a mixture or a mosaic of normal and mutant tissues. Because reproductive cells are not affected in a somatic mutation, therefore a somatic mutation cannot be transmitted to the progeny and may not be amenable to genetic analysis. Among the mutations that are most useful for genetic analysis are those whose effect can be turned on or off by the experimenter. These conditional mutations produce changes in phenotype in one set of environmental conditions called the restrictive conditions, but not in another set of conditions known as the permissive conditions. For example, a temperature-sensitive mutation is a conditional mutation whose expression depends on temperature. Usually the restrictive temperature is high. For example, in Drosophila it is 29 degrees centigrade, and the organism exhibits a mutant phenotype above the critical temperature. The permissive temperature is lower in Drosophila, which is 18 degrees centigrade, and under permissive conditions the phenotype is wild type or nearly wild type. Proteins containing amino acid replacements are often temperature sensitive. The protein folds properly and functions nearly normally under normal permissive conditions, but is unstable and denatures under restrictive conditions. Temperature sensitive amino acid replacements are frequently used to block particular biochemical pathways under restrictive conditions so as to test the importance of the pathway in various cellular processes such as DNA replication. An example of temperature sensitive mutation is found in the Siamese cat, as can be seen in the picture, with its black tipped paws, ears, and tail. In this breed, an enzyme in the pathway for the deposition of the black pigment melanin is temperature sensitive. The pathway is blocked at normal body temperature, and consequently the pigment is absent in the hair over most of the body where the temperature is warmest. The tips of the legs, ears, snout, and tail are cooler than the rest of the body, so the pigment is deposited in these areas. Mutations can also be classified according to the manner in which they affect the gene expression, and we gave it the title of classification by function. The major categories will be described here. Classical categories and terms were first introduced by Muller in 1931, but most of them have since been superseded by modern equivalents. Here we will use the modern terms for mutations. Replacements that inactivates the protein are known as loss of function mutations, and they are also known as null mutations or knockout mutations. Muller used to call it amorphic mutations. In loss of function mutation, what we observe is that a gene is inactivated, or the gene becomes completely non-functional. Such a mutation is known as loss of function mutations. Example for loss of function mutation may include a deletion of all or part of a gene and amino acid replacement that can inactivate the protein. A mutation that reduces but does not eliminate the level of expression of a gene or the activity of the gene product is called a hypomorphic mutation. Hypomorphic mutations typically results from nucleotide substitutions that prevent a protein from being produced at the normal level, or from an amino acid replacement that impairs protein function. This class of mutation is sometimes referred to as leaky. That is because the level of expression or activity differs from individual to individual by chance. A few individuals have enough enzyme activity to leak through to produce a normal phenotype. The opposite of a hypomorphic mutation is hypomorphic mutations. As the prefix hyper implies, a hypermorphic mutant produces a greater than normal level of gene expression, typically because the mutation changes the regulation of the gene so that the gene product is overproduced. Less commonly, an amino acid replacement may increase the activity of an enzyme or other protein. A gain of function mutation qualitatively alters the action of a gene. For example, a gain of function mutation may cause a gene to become active in a type of cell or tissue in which the gene is not normally active. Alternatively, it may result in the expression of a gene in development at a time during which the wild-type gene is not normally expressed. Whereas most loss of function and hypomorphic mutations are recessive, many gain of function mutations are dominant. Muller used to call these mutations as neomorphic mutations. Expression of a wild-type gene in an abnormal location is also called ectopic expression. For example, in Drosophila gene known as eyeless in tissue that do not normally form eyes result in the development of parts of compound eyes complete with eye pigments in abnormal locations. When we come to the molecular basis of mutation, we know that all mutations result from changes in the nucleotide sequence of DNA are from deletions, insertions, or rearrangement of DNA sequences in the genome. The major types of chromosomal rearrangements, as we discussed in the classroom, we know about it, that it also contains deletions, insertions, inversions, translocations. So here we will just talk about mutations at the molecular level. So first we'll come to nucleotide substitutions. As we know that we have 4 nucleotides in DNA, which are known as adenine, guanine, thymine, and cytosine. At the molecular level, the simplest type of mutations is a nucleotide substitutions, in which a nucleotide pair in a DNA duplex is replaced with a different nucleotide pair. For example, in adenine guanine substitution, an A is replaced with a guanine in one of the DNA strands. This substitutions temporarily creates a mismatched guanine and thymine base pair. At the very next replication, the mismatch is resolved as a proper GC base pair in one daughter molecule and as a proper A and T base pair in the other daughter molecule. In this case, the guanine and cytosine base pair is the mutant and the adenine and thymine base pair is the non-mutant. Similarly, in adenine-thymine substitution, an adenine is replaced with a thymine in one strand, creating a temporary thymine-thymine mismatch, which is resolved by replication as thymine-adenine in one daughter molecule and adenine-thymine in another. In this example, the thymine-adenine base pair is mutant and the adenine-thymine base pair is non-mutant. The thymine-adenine and the adenine-thymine are not equivalent, as can be seen by drawing it on a paper. And we know that adenine pairs to thymine and guanine pairs to cytosine. Some nucleotide substitution replace one pyrimidine base pair with the other one, or one purine base with the other. These are called transition mutations. The possible transition mutations are T to C or C to T, A to G or G to A. So there are possible 4 changes when it comes to transition mutations. Other nucleotide substitutions replace a pyrimidine with a purine or the other way around. These are called transversion mutations. The possible transversion mutations are 8 in number, which include T to A, T to G, C to A, C to G, A to T, A to C, G to T, and G to C. Altogether, there are 4 possible transitions and 8 possible transversions. If you write it on a paper, you will know that these are 4 and 8 in number. Therefore, if nucleotide substitutions were strictly random, then we would expect a 1:2 ratio of transitions to transversions. However, spontaneous nucleotide substitutions are often biased in favor of transitions. Among spontaneous nucleotide substitutions in the human genome, the ratio of transitions to transversions is approximately 2:1. Examination of the genetic code shows that the bias towards transition has an important implication for nucleotide substitutions in the 3rd position of codons. In all codons with a pyrimidine in the 3rd position, it does not matter which pyrimidine is present. Likewise, in most codons that end in a purine, either purine will do. As a consequence, most transition mutations in the 3rd codon positions do not change the amino acid in the encoded protein. Mutations that change the nucleotide sequence without changing the amino acid sequence are called synonymous mutations or silent mutations, because they change one codon into a synonym for the same amino acid. Mutational changes in nucleotides that are outside of coding region can also be silent. Most DNA polymorphism falls into this category. In non-coding regions, which include regulatory regions and the DNA between genes, the precise nucleotide sequence is often not critical. These sequences can undergo base substitutions, small deletions or additions or insertions of transposable elements and other rearrangements. Yet the mutations may have no detectable effect on the phenotype. Conversely, some non-coding sequences do have essential functions, and mutations in these sequences often do have phenotypic effects. As an example of missense mutation, we'll talk about sickle cell anemia. As we know that most nucleotide substitutions in coding regions do result in changed amino acids. These are called missense mutations or non-synonymous mutations. A change in the amino acid sequence of a protein may alter the biological property of the protein. The classic example of a phenotypic effect of a single amino acid change is the change responsible for the human hereditary disease known as sickle cell anemia. The molecular basis of the sickle cell mutation is shown in the figure. As can be seen in the figure, it consists of an adenine thymine to thymine adenine transversion in the second codon position for the sixth amino acid in the beta globin chain of hemoglobin, causing the normal glutamic acid to be replaced with valine. Thus, the amino acid replacement is denoted as either glutamic acid 6 to valine or E6V. When the sickle cell allele is homozygous, the defective beta polypeptide chain gives the hemoglobin protein a tendency to form long needle-like polymers. Red blood cells in which polymerization takes place become deformed into crescent sickle cell shapes. Some of these deformed red blood cells are destroyed immediately, reducing the oxygen carrying capacity of the blood and causing the anemia, whereas others become trapped in capillary vessels and cause partial or complete blocks in capillary circulation. The consequence of this mutation or replacement are a set of pleiotropic effects. All of these effects are related to the breakdown of red blood cells, to the decreased oxygen carrying capacity of the blood, or to the physiological adjustments the body makes to try to compensate for the disease, such as enlargement of the spleen. Patients with sickle cell anemia suffers bouts of severe pain. The anemia causes impaired growth, weakness, and jaundice. Affected people are so generally weakened that they are susceptible to bacterial infections, which are the most common cause of death in the children with the disease. Although sickle cell anemia is a severe genetic disease that often results in premature death, it is relatively frequent in areas of Africa and the Middle East, in which a type of malaria caused by the protozoan parasite Plasmodium falciparum is widespread. The association between sickle cell anemia and malaria is not coincidental. It results from the ability of the mutant beta hemoglobin to afford some protection against the malarial infection. As part of its life cycle, the parasite Plasmodium falciparum passes from a mosquito to a human through the mosquito bite. The initial stages of infection take place in the cell in the liver, where specialized forms of the parasite that are able to infect and multiply in the red blood cells are produced. Widespread infection of red blood cells impairs the ability of the blood to carry oxygen, causing the weakness, anemia, and the jaundice characteristics of malaria. However, in people who are heterozygous for the sickle cell allele, infection with malaria is less likely and also less severe. The partial resistance offered by the heterozygous sickle cell genotype creates a genetic balancing act between the prevalence of the genetic disease sickle cell anemia and the prevalence of the parasitic disease malaria. If the mutant sickle cell hemoglobins becomes too frequent, more lives will be lost from sickle cell anemia than are gained by the protection against malaria. Conversely, if the mutant sickle cell hemoglobins become too rare, fewer lives will be lost from sickle cell anemia, but the gain is offset by more deaths from malaria. The end result of this kind of genetic balancing acts is discussed in quantitative terms. In contrast to the glutamic acid valine replacement, some amino acid replacements in hemoglobin have negligible effects on phenotype. Amino acid replacements are often tolerated in other proteins as well. The key issue is whether a particular amino acid replacement affects protein folding, function, or stability. And this outcome depends on the protein, the amino acid, and the precise role in the structure and function of the protein and the nature of the amino acid replacement. For example, the replacement of one hydrophilic amino acid with another is expected to have a lesser effect than replacement of a hydrophilic amino acid with a hydrophobic amino acid. Any change in the active site of an enzyme usually decreases the activity of the enzyme. As can be seen in the figure on the left side, which is for results of a single base change, this figure illustrates the 9 possible codons that can result from a single nucleotide substitution in the UAU codon for tyrosine, organized according to whether the first, second, or third nucleotide in the codon is mutant. One mutation is silent, which is in the box, and 6 missense mutations that changes the amino acid inserted in the polypeptide chain. The other 2 mutations create a stop codon that results in the premature termination of translation and the production of a truncated polypeptide. A nucleotide substitution that creates a new stop codon is known as nonsense mutation. Because nonsense mutation causes premature chain termination, the remaining polypeptide fragment is nearly always non-functional. When it comes to insertions or deletions of nucleotides, we know that it also occurs in DNA at a considerably lower rate than nucleotide substitution. Most insertions and deletions in mammalian genomes are small, which are typically 1 to 6 base pairs, but some are large enough to eliminate entire genes or part of a chromosome. If they occur outside of the coding region, small insertions or deletions may have negligible effects on phenotype unless they affect the expression of a nearby genes. Within a coding region, the insertion or deletion of exactly 3 nucleotides result in the addition or elimination of one amino acid from the encoded polypeptide chain, because 3 nucleotide is the length of a codon. An insertion or deletion of 6 nucleotides aids or eliminates 2 amino acids, and an insertion or deletion of 9 nucleotides aids or eliminates 3 amino acids. In general, provided that the number of nucleotides inserted or deleted an exact multiple of 3, an insertion or deletion aids or eliminates some number of amino acids from the polypeptide chain. If the length of an insertion or deletion is not an exact multiple of 3 nucleotides, the mutation shifts the phase in which the ribosome reads the triplet codons and consequently alters the amino acid downstream from the site of the mutation. Such mutations are called frame shift mutations because they shift the reading frame of the codon in the messenger RNA. A common type of frame shift mutation is a single nucleotide insertion or deletion. An example of a single nucleotide insertion in the human beta globin gene is shown in the figure, where In the figure, A is the wild type sequence and B is the mutant sequence with a cytosine to guanine nucleotide pair inserted at the position of the sunburst. The result of the insertion is that, starting with the codon in which the insertion occurs, the entire amino acid sequence of the polypeptide is altered because the reading frame is shifted in the example by +1. Translation continues in the shifted reading frame until a termination codon is encountered. Translation of a completely random nucleotide sequence would be expected to continue for only approximately 20 amino acids before running into a termination codon. Any insertion or deletion that is not a multiple of 3 nucleotides results in a shifted reading frame, and all amino acids downstream from the insertion are different from the original. Unless it occurs very near the carboxyl terminus of protein, a frame shift mutation almost always results in the synthesis of a non-functional protein. Now we come to dynamic mutation of trinucleotide repeats. The genetic studies of an X-linked form of intellectual disability revealed an unexpected class of mutation dubbed as dynamic mutations. They are called dynamic mutations because of the extraordinary genetic instability of the region of the DNA involved. The X-linked conditions, which is one of at least 12 genetic disorders associated with dynamic mutations, is associated with a class of X chromosomes that tends to fracture in cultured cells that are stored for DNA precursors such as nucleotides. The position of the fracture is in the region XQ27.3 near the end of the long arm of the chromosome. The X chromosome containing the site are called fragile X chromosomes, and the associated form of intellectual disability is known as fragile X syndrome. Fragile X syndrome affects approximately 1 in 2,500 males and 1 in 5,000 females. It accounts for nearly half of all cases of X-linked intellectual disability and is second only to Down syndrome as a cause of inherited mental impairment. Fragile X syndrome is highly variable in severity. Males are usually more severely affected than females. Developmental delays in speech and communication skills are common, as well as delays in such gross motor skills as sitting up and learning to walk. Physical symptoms of this syndrome may include a long face with protruding ears, weakness in connective tissue resulting in poor muscular tone, and extremely flexible joints, and enlarged testicles in males past puberty. Intellectual disability is usually moderate in males and mild in females. Behavioral effects may include anxiety, poor concentration, trouble coping with sensory stimuli, avoidance of eye contact, and tantrums of emotional outbursts. These symptoms are non-specific and overlap with such conditions as autism and attention deficit hyperactivity disorder. A hint of something unusual about Fragile X syndrome was the paradoxical pattern of its inheritance. Approximately 1 in 5 males who carry the Fragile X chromosome are themselves phenotypically normal and also have phenotypically normal children. The oddity is that the heterozygous daughters of such a transmitting male often have affected children of both sexes. The transmitting male is sometimes not affected, but the X chromosomes that he transmits to his daughters somehow becomes altered in the female in such a way that sons and daughters in the next generation are affected. Both affected and normal granddaughters of the transmitting male may have affected progeny. The molecular basis of the fragile X chromosome has been traced to a trinucleotide repeat of the form CGG or equivalently CCG, which is cytosine, and guanine on the other strand. Normal X chromosomes have 6 to 54 tandem copies of the repeating unit with an average of about 30, whereas affected persons have 230 to 2300 or more copies of the repeat. The trinucleotide repeat in the X chromosome in transmitting males is called the pre-mutation and has an intermediate number of copies ranging from 52 to 230. Approximately 1 in 250 females and 1 in 800 males carry an X chromosome with the pre-mutation. The unprecedented feature of the trinucleotide pre-mutation is that when transmitted by females and only by the females, it often increases in copy number, a phenomenon called trinucleotide expansion, to 230 copies or more, at which stage the chromosome causes fragile X syndrome. The amplification does not take place in transmission through a male. The functional basis of fragile X syndrome is that an excessive number of copies of the CGG repeat causes loss of function of a gene designated as FMR1, which is known as fragile site mental retardation 1, in which the CGG repeat is present. Most patients with fragile X syndrome exhibit no FMR1 messenger RNA, whereas average persons and carriers do show expression. The FMR1 gene is expressed primarily in the brain and testis, which explains the strange association between mental and testis abnormalities in affected males. There is approximately an 80% chance that a pre-mutation transmitted by a female will undergo amplification. Surprisingly, the amplification does not take place in the mother's germline, but rather in somatic cells of the early embryo. Amplification occurs to a different extent in different somatic cells, so individuals with fragile X syndrome are somatic mosaics for cells with different numbers of copies of the CGG repeat in the X chromosome. That accounts for the great variation in the severity of the fragile X syndrome from one affected individual to the next. Other genetic diseases associated with dynamic mutations include the neurological disorder myotonic dystrophy, which is at chromosome 19 with an unstable repeat of CTG, Kennedy disease on the X chromosome, a repeat of AGC, Friedreich ataxia, which is on chromosome 9 with a repeat of AAG, spinocerebellar ataxia type 1, which is on chromosome 6 with a repeat of AGC, and Huntington disease on chromosome 4 with a repeat of AGC. With fragile X syndrome and myotonic dystrophy, the trinucleotide expansion occurs primarily or exclusively when transmitted by females. In contrast with spinocerebellar ataxia type 1 and Huntington disease, they occur primarily or exclusively when transmitted by males. Some trinucleotide repeats can undergo amplification when transmitted by either sex. Some dynamic mutations also exhibit a phenomenon called anticipation. In anticipation, the number of repeats gradually increases as the trinucleotide repeat is transmitted through several generations. This results in a more severe disease with a progressively earlier age of onset through time. The molecular mechanism of trinucleotide expansion is through a process which is known as replication slippage or slipped strand mispairing.